My name is Alexey Danilov. I'm Director for Early Phase Therapeutics and Professor in the Department of Hematology at City of Hope. At this conference, we presented early data on two different BTK degraders, NX-2127 and NX-5948. BTK degraders is a novel class of agents, and you probably heard about BTK inhibitors such as ibrutinib, acalabrutinib, and zanubrutinib. And BTK degraders are actually different in a sense that BTK inhibitors block the activity, the function of the molecule BTK, which lymphoma cells and CLL cells heavily rely on in their survival and proliferation, meaning they depend on this particular kinase protein to make new cells. So BTK inhibitors block the activity or the function of the kinase. But meanwhile, BTK degraders fully obliterate or destroy the kinase and pull it up into pieces by linking it to the cell waste processing machinery. Why is it important? The problem is that even though BTK inhibitors can work for a very long time, eventually patients develop resistance. So the BTK protein learns how to adapt to the presence of these molecules. It introduces mutations so that these molecules no longer can bind well to the kinase. And as a result, CLL cells or lymphoma cells become resistant. So BTK degraders, however, work in a setting when those different types of mutations are present, because it doesn't just bind the functional pocket of the kinase, it destroys it completely. There are several trials going on. The two BTK degraders that we investigated in the study that we presented at this ASH, NX-2127 and NX-5948, are currently in phase 1 clinical trials. And some of the results will be presented at this ASH meeting as well. The data looks really good. The response rate is about 70%, meaning that at least 70% of patients achieve a formal response where their lymph nodes decrease, where their anemia improves, and they feel better. But virtually every patient derives clinical benefit. There are also other BTK degraders in development. One BTK degrader is developed by BeiGene. I believe it's BGB-16553. And there is also a BTK degrader developed by AbbVie, ABBV-101. And they are also in clinical trials enrolling patients. Most of these trials are larger scale. There are large international trials. They're open at many sites in the United States, United Kingdom, and continental Europe. That's where I'm not aware of any other sites outside of these continents, but these are certainly global studies in that sense. Australia, actually. As this data is still early, the initial space where they will be used is relapsed refractory. So right now, patients who can go on these studies typically would have progressed on a BTK inhibitor and potentially also on BCL2 inhibitor venetoclax. But certainly there are plans to introduce, to study these molecules in earlier lines of therapy. So I will say that these are completely new drugs, state-of-the-art technology, which are very different from all the small molecules that we have currently working in the clinic. And in general, there have been very good, tremendous advances in treatment of CLL with very many effective therapies. And this is another wonderful option. I have patients who have been on these drugs, successfully treated for about 3 years now, who have progressed on multiple therapies. So these are very effective agents, and I would highly recommend to seek out these clinical trials. So there's been quite a lot of new good information at this ASH. The Amplify study results have been presented, which will solidify the place of a novel doublet, acalabrutinib and venetoclax, in patients with previously untreated CLL. We are also presenting early results from a phase 1 trial of a bispecific antibody, epcoritamab, in relapsed refractory CLL. This is also a completely new mechanism of action where we attack the CLL cell, but at the same time engage the patient's immune system through their T cells specifically to kill the CLL cells. And the results there are also spectacular.